So with glycogen metabolism, we're going to talk about it two different ways, as with most things we do. First, we're going to describe how we degrade glycogen, and then we'll describe how we make it. So the first process is a catabolic process where we degrade a molecule to release small pieces of it that we can use for other reactions, or in this case as glucose for fuel. And in the second half of the lecture, we're going to briefly talk about how we take glucose and make a glycogen molecule when we like to store glucose. But first off, what is glycogen? So glycogen is basically a storage form of glucose, right? It's not a fatty acid, it's not a protein, although it has a very small protein beginning, but it's mostly made of glucose. And so the bottom right is a picture of a glycogen granule, obviously not to scale, but you see that little yellow sphere in the center? That's an icon representing the protein that's used to get this granule started. Right? It's called glycogen. It's a very small protein. And at some point, we start adding sugars, glucose molecules, to this protein. And then the thing balloons from there. We add more sugars, and they become long chains. You see the little sticks in the picture. And if I only did that, it would make one really long stick. So at some point, we have to start branching. So the sticks get branched. So at one point, 2 sugars would be attached at one point. And then it'll branch out farther and get longer. And then we branch it again and again and again. So the more branching we add to this is mainly the difference between the glycogen found in humans versus the glycogen found in dogs or cats or mice or trees or bacteria or any other place, or plants. So the other name we call glycogen is, sometimes you'll call it starch if it's found from a plant source. You'll call it amylose or amylopectin, or even sometimes it's just referred to as storage sugar or stored sugars. Right? So all those represent the same thing, and we're going to refer to it as glycogen. And it's just a bunch of glucose put together, and we're going to talk about how it's disassembled first. Okay, so where is glycogen in your body? Well, every cell has some. Some organs have a lot more than others. So the liver is about 10% glycogen by weight, right, which is a lot. So if you think about an average cell, most of the mass of a cell is going to be water, right? And then you've got proteins and some lipids, and then very little is actually just sugar inside the cell, because it's not a solid mass of sugar. But in the case of the liver, 10% of its mass is made of glycogen, or made of glucose. So there's a large store of glucose in the liver. Why do we do that? Because the liver is selfless. It's not interested in its own well-being as much as it is determined to maintain a blood glucose concentration. So its goal is to store glucose in this way and then release it as the body needs. So between meals, because you're not constantly eating. The other major storage site for glycogen is in skeletal muscle. And here it's only about 2% by weight. So again, mostly water again, a lot more protein here. We have some lipids, and we also have a lot less glycogen storage than we had in the liver. What's the point of this glycogen storage? Well, muscles are very selfish. The glycogen here is for its own needs, right? It's not going to export it for other cells' use. Once glucose is inside of a muscle cell, it's for that cell. So they need to store it because you never know when you're going to need to run. And so if I ask a question of which contains more glycogen, liver or muscles, well, by the organ themselves, looking at individual organs like a leg muscle versus a liver, the liver's got more, it's 10% versus 2% in the muscle. But overall, which organ holds more glycogen in your body? Well, there's far more muscle mass than there is liver mass. And even though it's 1/5 the concentration, there's way more muscle mass than there is liver in your average person. And in athletes, far more. Right? So there's far more glycogen stored in muscle by total count than there is just the liver. But the liver is more percentage-wise glycogen than the muscles are. And the last question is, where is the glycogen stored? It's stored in the cytoplasm of the cell. So that big granule you see on the right is just packaged in a closet in the cytoplasm until we need it. So here's a more detailed picture of how glycogen is put together. So if you remember our structure of glucose when we did carbohydrates, glucose can wrap around into its hemiacetal form or its ring form, And we generally drew that this way, as you see on the picture here. Here's 6 glucose molecules hooked together, and the chain indicates that it's a long polymer. It keeps going. So if you look at the bottom row, it shows 4 glucose molecules there hooked together from one end to the other, and it's an alpha 1,4 glycosidic bond. Remember how we got that number? It's connecting number 1 of one sugar to number 4 of the next. And number 1 was our new chiral center, right? So it could be up or down. It could be beta or alpha, respectively. In this case, it's down, so we call it alpha 1,4. And you can see that can make a really long chain if I keep going. Those are represented in our little model by the sticks. Occasionally, we'll have an alpha 1, not instead connected to a 4, but connected to the 6 somewhere else in the chain. So it's a branch point. That's that alpha 1,6 linkage you see up there. So if we keep branching every so often, our long chain becomes a more compact mass, right? So it can take on a more spherical form like you see in the right, which makes it a lot more soluble, right? Or more compact to stay in the cell. For a really long chain without branches, it would not be soluble at all. It would be a long fiber, right? Much like actin molecules are. So how do we degrade this glycogen? Well, we're going to have to do a few things to it. We're going to cut off one of the sugars, right? So this big, long chain of sugars, we're going to just cut one off. We'll see how that's done. At some point, we won't be able to reach the next sugar because of our branching issues. You can imagine if I'm chopping down one long chain and I come to that branch point, I can't easily remove the next one. I'm going to have to deal with that branch. So I'm going to remodel this thing a little bit. I'm going to move some sugars around to free up that branch point. And then finally, my sugar that I get off, that I removed, is not just plain old glucose all the time. For the most part, it's going to be a phosphate already attached. We'll talk about that. And it's in the form of glucose 1-phosphate. And if you remember from glycolysis, there is no glucose 1-phosphate. The phosphate was on number 6. So in order to get this ready for glycolysis, we're going to have to move that phosphate from number 1 to number 6. And that's going to be a simple isomerase. Right, so we'll move it to number 6, and then it's ready to enter glycolysis at step 2. Okay. And so if we start chopping this down, eventually we'll get to the point where the only thing left will be the core, that little protein, with a few sugars still attached that I can't remove. And that's called limit dextran. We'll talk about it in a few minutes too. And then if I want to degrade more, I'm going to have to find another source of sugar. Or alternatively, if I have a broken down and I want to store it later, build more onto it, I can build onto those granules again. So they're not consumed completely. You can always build and remove from them as you need. Okay. If your muscles with that 2% body mass of glycogen, 2% mass of glycogen runs out, then your muscle cells are going to need another source of glucose. And hopefully at that point, the liver's detected that the muscles are working hard and I need to send some more glucose their way. So the liver is going to degrade its massive store of glycogen and export it for muscles to use. That's a little slower getting into the cells and being used than the muscles using its own in-closet or in-house supply. So you can't work as hard, or muscles work as fast. And runners will call this hitting the wall, marathon runners, where they have to switch from only the energy immediately available in the muscle from glycogen to using the glycogen available from the liver, or switching to a lipid source. Okay. To illustrate that, I have a little graph for you. So it's a graph I found, I don't know, quite a while ago. I'm sure these times are not the world records anymore. It's been updated. But these represent, how fast humans can do certain things. So if your running time is only over 100 meters, we can do that, not you or I, but Usain Bolt and his company can run these fairly fast, under 10 seconds, which equates to something like 10.2 meters per second or 23 miles an hour. Can we keep that up indefinitely? Of course not. Right? And we'll see why in a second. If you run for a longer race, say a kilometer, they call it a sprint here, but it's a 1 kilometer run, and it's done in a little over 2 minutes. You or I, probably a lot longer than that, but a little over 2 minutes for these world-class athletes, and that's about 7.5 meters per second, or 17 or so miles an hour. That's quite a bit slower than the pace for the short distance run. And if you move on to a much longer distance, and of course, if you notice, the running time here is on a log scale, so this gets quite large rapidly. So if you run your marathon of approximately 42,000 meters, it takes a little over 2 hours for these athletes to run that, and the speed is dropped to about half of what our sprinting speed is. Right? And I found another point. I wanted another longer piece on this graph, so I found an endurance race where you run for 24 hours straight. How far can you run? Much like the Le Mans for racing, but it's humans instead of cars. How far can you run in 24 hours? A little over 300 kilometers, which is a long way. But it's slowed down on the average speed again. You're only at 3.5 meters per second, or just under 8 miles an hour, which is not much faster than walking pace. Okay, so if you look at this graph, it almost looks straight, like you could draw a straight line through all these points, but more subtly, there's some different slopes you can get out of this. So in the beginning, when you have the 100 meter or the 200 meter sprints, it doesn't look like the time drops off that much, or the speed drops off that much. So to round things off crudely, for a 100 meter sprint, let's say it takes 10 seconds. That would be 10 meters per second. For a 200 meter race, let's say it takes 20 seconds. Right? It takes a little longer than on average than the 10 meter pace, but again, that's around 10 meters per second. So the pace hasn't dropped off after the first 10, 20, 25 seconds or so. And then it starts to decline. Once you get up to a little over 2 minutes, you're down to a lot slower average speed. What's happening? So if you look at the bottom, I put a little black dot by the beginning there. That's our available ATP in the muscles. You will go through that within seconds when you start running at this pace. But you don't run out immediately. There's another source of phosphate nearby. It's called creatine. We haven't talked about the structure of creatine, but think of it as another reservoir of high-energy phosphates available. So when ATP runs out in the muscle, creatine can supply ATP for about the same amount of time again. You can keep running a little longer. It doesn't seem like that much of a benefit. You can run for, say 24 seconds instead of 12. Well, it doesn't seem like a huge advantage. Well, that's an extra 12 seconds to get away from the predator. Right? So if you can get to that safe haven or whatever, the bear can't get you, or the lion can't get you. And it might be the difference between life and death. And the next part, running for much longer, several minutes, that's more of a reserve for exercise, maybe going from place to place. It's not so much evading a predator in those kind of terms. So if you look at those next few points, I drew a red line through it for you. It gives a slope of decreasing performance there. And this is the muscle's glycogen that's available, right? That 2% we talked about. That 2% glycogen being converted to lactate initially, that's fermentation, so you can continue doing glycolysis. Eventually, we'll be more efficient at it and convert it to CO2 with the citric acid cycle, and you need a lot of mitochondria to do that. So if the muscles aren't in shape, you won't be able to get to this quickly. Athletes will have more mitochondria. And so you get a decrease in the performance, but it's not as bad as later. And you notice the maximum ATP production, it's not 70 something like it was before. It's dropped off in the 40s and 20s and teens. Right? But there's much more phosphate available, so we can extend doing this for a much longer time frame. And then finally, we have that hitting the wall. We run out of muscle glycogen, and now we're dependent on the liver to supply glycogen, or we're getting energy from degradation of fats and other materials. So that's this next line, this blue slope. It's a lot shallower than it was before. Right? So it's a slower delivery of phosphates, but the pool or the reservoir of phosphates is enormous compared to what we had before. So you could continue this for hours, right? In one case here, up to 24 hours. Right? So at that point, what happens? How far can we go? How much longer can we run at this pace? Well, it doesn't become an energy, a matter of fuel availability at that point, because we have 4 million available phosphates there. You could run this for several days on that. The problem is you're going to run into fatigue in the structures, right? Not so much the energy supply, but the tendons, the bones, the muscles, the skin, everything else is going to start to cause, excessive damage to those tissues. So you're going to have some failure of a part, not so much a fuel source. So if you put it in terms of the car racing, it's not so much you run out of fuel, but mechanically it starts to fall apart, right? Parts of the vehicle can't stand that much rigorous use over that amount of time. Okay, so to take glycogen apart, we're going to do it in 4 parts, right? 4 steps. So again, here's a glycogen molecule at the top. I reworked the figure a little bit from the one I posted for you. If you notice on the first part here, where the oxygen bridging the 2 sugars, in the original figure, they like to show this with a little angle bar, right? Showing you that the there appears to be an extra carbon there, and that's not true. There isn't an extra carbon. What they're trying to show you is that that oxygen on the left was pointed down, or alpha, and the oxygen connecting to the second sugar was also down, because down is right for number 4 on glucose. So I rearranged it to show you they're both down still, but with straight lines, because there is no additional carbon there. I don't want to confuse you. Okay, so we have glycogen connected in a long, long chain here. And the very last sugar added, you notice the sugar on the left of the pair is just an OH on the end, right? It's the very last sugar in the chain, whereas the other one has an R written there, which means it's part of a longer chain. So if you scroll back to the other figure, we're taking apart the sugar on the very end of the chain here, where it could be part of a much longer chain. Right, so we have this glycogen bond sitting here, this glycosidic bond sitting here, and it's connecting the number 1 carbon to the number 4 of the next sugar. Right, we're going to use an enzyme called glycogen phosphorylase, and it is a hydrolase in the class of enzymes, but keep in mind we talked about hydrolases don't necessarily use water to break a bond. That's the most common way to do it, right? Regular old hydrolase uses a water molecule to break a bond. One side gets H, one side gets OH, and you're in 2 pieces. No big deal. You don't have to use water. We can use other small molecules like ammonia, or in this case, phosphate. Right? So when it's a phosphate being used as the saw, right? We're not using a water saw here, it's a phosphate saw. One side again gets H, and the other side gets the remainder of the phosphate. So that's shown in the reddish color on the right, where I broke the bond, and the sugar on the right got an H, and the sugar on the left that I'm cutting off gets the remainder of the phosphate, the PO3. Okay, so this enzyme is known as glycogen phosphorylase, and all phosphorylases are kinases that use phosphate as the saw. Right? So if you think of it as a simple analogy, sharks are fish, but not all fish are sharks. Right? So in this case, the shark is the phosphorylase, but the kinase, sorry, not the kinase, the hydrolase, the larger category would be fish. So let me say that again in case you're confused. So a phosphorylase is a member of a larger family of hydrolases. It's the one that specifically uses phosphate. Hydrolases can use lots of things as saws: water, phosphate, ammonia. But the ones that use phosphate are called phosphorylases. It's much like a shark is a subgroup of fish. Okay? So how do we do that? We add the orthophosphate, abbreviated PI, or if you see it in red above, it's the HPO4 with the -2 charge. You add it to it, you break the bond, one side gets H, that's the remaining glycogen chain that's now 1 glucose shorter, and the released molecule is not just glucose, but glucose with a phosphate on it. And you notice from the position, it's on number 1. So it's called glucose 1-phosphate. Now we can keep doing this over and over. I could take the glycogen that is now 1 glucose shorter, apply the same enzyme with another phosphate, and cut off the next glucose, and the next, and the next, and the next. Where does this end? Well, I can only cut so far until I get close to one of those branch points. Okay, so I'm removing glucose as glucose 1-phosphate many times until I get close enough to a branch point that the enzyme just can't reach the bond. It's physically unable to reach the bond it wants to cut. When we get to that point, it's called limit dextran. Right? So when the enzyme cannot reach the next bond, I can't cut the next 1,4 bond, it's called limit dextran. And we need to do some rearranging at that point. So I have a diagram here on the right that kind of explains what you can and cannot cut off at this moment. So what this little cartoon is showing is each of the little hexagons represents a glucose, and they're all connected in alpha 1,4 bonds, except for that one point you see alpha 1,6, where it's connected to number 6 sticking up. And my glycogen phosphorylase, this first enzyme we talked about, can cut off all the pink colored sugars. Okay, every one of those on the right-hand side or at the end of the chain is exactly what we just described. The enzyme binds, uses a phosphate, represented by PI here, to break the bond between those sugars, releasing it as glucose 1-phosphate. And I can cut off all 8 of those pink sugars at this point. Right? It's not always 8. Just in this diagram, in this example, there were 8 that were accessible. What do you notice about the ones that are left? Well, from the branch point, I can't get any closer than 4. Right? So what do I mean by branch point? Branch point, I mean the sugar that's connected to 3 other sugars. In a normal chain, every sugar except the one on the end is connected to the ones on opposite sides of it. So just like in a carbon chain, right? Every carbon's connected to the 2 around it except the one on the end. So here we have all these sugars connected to 2 other sugars except this one, right? So this one's connected to 3 other sugars, the one before it, the one after it by 1,4 linkages, and then the one connected by 1,6. It's the only sugar up there that has bonds to 3 other sugars. Okay, so from that point, I'm going to count 4 sugars away. I'm not going to count that one as number 1, right? So it's not inclusive counting. But after that, I'm going to count to 4. So if I count 1, 2, 3, 4 yellows before I arrive at a pink one. On the top part, I'll count 1, that's the green one, and then 2, 3, 4 in blue, and then I can't get any closer than that. So that's why all of those pink ones could be removed because they're number 5 or onward. Okay, so I can cut those away with phosphorylase, no problem. I get 8, in this example, glucose 1-phosphates. Then I arrive at that limit dextran. I can't cut any more off without some rearranging here. So the next enzyme we'll talk about has 2 features. It accomplishes 2 pieces of chemistry, right? It's the same enzyme, but it accomplishes 2 goals. Okay, it's called the debranching enzyme, or the 1,6-glucosidase, and we'll see what it does. It has 2 features. We're going to take the 3 blue ones from the top strand, because that's the strand I'd like to debranch and get rid of that branch point. I'm going to take the 3 blue ones, it's always 3, pluck them off, and reattach them to the other strand. So these 3 blues are removed from the green one, right, and then reattached to the end of the yellow chain, which would be here. Okay. What type of enzyme would that be? Well, I took something away from this molecule, and then I gave it right back to the molecule in a different way. So what type of enzyme would this be?
Isomerase.
Isomerase, exactly. When they discovered this enzyme, they didn't know it had these 2 features. There were 2 independent groups studying this, and one discovered the feature where it removes this piece, and the other discovered the feature where the very same enzyme is going to cut off that green one. Right? So when they only discovered that it removed this piece, unfortunately it got the name glycogen transferase, which is misleading, and that yes, it is transferring something, I agree, but it doesn't fit the definition of a transferase, because a transferase gives it to a new molecule. It put it right back on the same molecule in a different spot. So like you just said, that is an isomerase. Fortunately, the other group that was studying this recognized that the same enzyme cuts off the green one. Cutting off the green one would be what type of enzyme? Just simply cutting it off. And in fact, you see it uses water here. So what class would the action of cutting off that green sugar be? So I'm taking a molecule, adding water, and removing a piece. Very similar to something we just did. All right, can you see it's also a hydrolase? Just like the first enzyme we talked about. If I cut off the green sugar, and not using phosphate this time, I'm just using plain old water. So it's not a phosphorylase, but it is a hydrolase. So it's another fish. It's not a shark this time. Maybe it's a tuna. All right, so I'm cutting off the green one using water, makes it a hydrolase. But fortunately, those 2 actions were found out to be part of the very same enzyme. It does both of those things. So we can fortunately lose that terrible name called transferase, because it was in fact not a transferase, and we just call the thing the debranching enzyme now. It adopted the name of the second one. So it moves the 3 out of the way. It has an isomerase feature, and then it has a feature of cutting off the green one, a hydrolase feature. So it just debranches the thing. And you see when we're done, we have a chain that is no longer branched. So at this point, our enzyme number 1 can come back along and start chopping off these sugars, the 3 blue ones, and then some of the yellow ones, right? And have to stop at some point when I get within 4 of the core. For the same reason I can't get within 4 of the branch point, I can't get within 4 of the core protein, because it just can't reach the bond. Okay, so we've only used 2 enzymes so far. The first enzyme removes whatever's available that it can reach, the pink ones at the top. Then when I get to that limit dextran where I can't remove anymore with the phosphorylase, I have my debranching enzyme remodel some things for me, right? It moves the 3 onto another strand, it cuts off the green one, and then I can apply the first enzyme again and chop it down until there's only 4 left. If there were multiple branching sites, we just repeat the process until we can get down to just 4 left. And that's as much as you can remove from a glycogen granule without destroying the core. Okay? The key difference here is when phosphorylase, glycogen phosphorylase, it's shortened to just called phosphorylase sometimes, removes the sugars, they're removed as glucose 1-phosphates. In that case, we used a phosphate to break the bond, right? It ends up on the sugar. In the case of the debranching enzyme, removing that green one, I just used plain old water. So when it comes off as the green sugar, it's just plain glucose. Well, the glucose 1-phosphate is not quite ready to enter glycolysis. What the green one is. All I have to do is give it to hexokinase. It'll put a phosphate on number 6 and we're good to go. The glucose with the phosphate on number 1 is not quite ready yet. We need to move it to number 6. So that introduces our third enzyme, the phosphoglucomutase. The name makes sense because it is an isomerase. It's going to move the phosphate from number 1 to number 6. And isomerases that move things bigger than, say, hydrogen, like it could move a methyl group, it could move an amino group, it could move a phosphate in this case, it could move something even larger. When it moves something larger than a hydrogen, we don't call it just an isomerase, more specifically we call it a mutase. So again, like the fish example, all mutases are isomerases. They just move something bigger than hydrogen. So if all you did was rearrange a double bond in a molecule by moving a couple protons around, that's not a mutase because it didn't move something big. It has to move something of significant size, bigger than a hydrogen. But if you moved a phosphate around, or if you moved an entire carbon chain around, or if you moved a amino group around, or carboxyl group, whatever it might be, then you can call it a mutase. So that describes the mutase part of the name. What is it mutating? What is it moving? Well, it's moving a part of an already phosphorylated glucose, a phosphoglucose. So it's called phosphoglucomutase. Don't confuse it with the phosphoglucose isomerase, our second step in glycolysis. That one is not a mutase because all it moves is a hydrogen, right? It's moving the double bond, but all it's really moving are hydrogens and electrons. Okay, so we're going to move the phosphate that's on number 1 over to number 6. How does this enzyme do this? Well, it's rather simple. This depiction here, the yellow swirly line there represents the enzyme itself and its active site. The serine at the top is one of its amino acids, and it starts out with a phosphate on the serine, a little red phosphate up there. The glucose 1-phosphate shows up, it's the substrate. It has a phosphate on number 1, shown in blue, and nothing on number 6. And the enzyme donates its red phosphate onto number 6. So at this point, we have this transition state. The enzyme's not finished, but it has this transition state where the glucose has 2 phosphates on it. At that point, the glucose with its double phosphates flips over in the active site, where 1 sits where 6 was, and 6 sits where the molecule just flips over, just does a tumble. And then the one on number 1 gets transported onto the serine that just lost 1. So the blue one has taken the place of the pink on the enzyme. And then the glucose 6-phosphate can leave. And the enzyme is no worse for wear. One phosphate's no different or no worse than another. So they can do this reaction again with another glucose 1-phosphate. Of course, you can imagine this going completely backwards. A glucose 6-phosphate shows up, gets the phosphate in blue attached to it, right, at number 1, flips over, and puts the phosphate from number 6 back on the serine, leaving as glucose 1-phosphate. So totally reversible. What would determine which way this enzyme is going to go? Could you imagine it's delta G? It's probably not going to be a very big number. These molecules are almost identical. So it's totally reversible. Which way it goes is depend on the concentrations of glucose 1-phosphate and glucose 6-phosphate in its vicinity. If there's lots of glucose 1-phosphate, we're degrading glycogen like crazy, like during exercise, right? is going to go to the right. There's going to be plenty more glucose 1-phosphate around than glucose 6 because we're using it in glycolysis. However, if you're in couch potato mode and you've been eating a lot of sugars and your liver is going, wow, we got a lot of sugar around, let's store it as glycogen for later. We're going to take the glucose 6-phosphate, which is in far excess compared to glucose 1. We don't need to do glycolysis right now. And let's store it as glycogen, so we'll turn it into glucose 1-phosphate. So this enzyme really isn't a control mechanism. It just goes with the flow. Whichever direction needs to go, that's the direction it will go. Okay? The difference here is between muscles and liver. If you're a liver cell, the reason to degrade glycogen is so you can export it as glucose for the rest of the body. That glucose 6-phosphate sitting there is going to be difficult to export out of the cell because of that negative charge on it, right? So in order to export it, we have to cut off the phosphate, right? So cutting off the phosphate, we use an enzyme called a phosphatase, right, which is yet another hydrolase. And all it does is use water to hack off that phosphate, right? And we end up with just plain old glucose, and the liver can export the glucose. In every other cell type, a little bit goes on in the kidney too, but for the most part, just liver does that. But in every other cell type, mainly muscles, we get to glucose 6-phosphate and we never let that glucose go. Remember, muscle cells are selfish. So if a glucose gets in their cell, they don't ever let it go. They may store it as glycogen and release it later as glucose 6-phosphate in the cytoplasm, but we never export it. At this point, we don't want it to be cut off. We want to go through glycolysis. So we're going to turn it into fructose 6-phosphate and so forth the rest of the way down glycolysis. So only liver is going to export that glucose as glucose. All right, here's the enzyme that did the first step that we talked about. Remember we called it glycogen phosphorylase? I had it listed as number 1 a few slides ago. We're just going to refer to it as phosphorylase because that's its common name. And it exists in a couple forms, right? There's one in the liver, there's one in muscles, and they act quite a bit differently. And I don't want you to learn all the details of it. I'll save that for another class. But what would the liver be interested in? If you're a liver cell, if you're the liver organ, the reason to degrade glycogen would be because the rest of the body needs it. The blood glucose has gone down, right? So the liver's phosphorylase is sensitive to the concentration of glucose. Whereas a muscle cell doesn't really care about the glucose in the blood other than it better be there. So it doesn't really care about exporting its own sugars. So the muscle's phosphorylase only cares about the state of that muscle cell, which it determines if it needs to degrade its glycogen or not by the presence or absence of ATP. So if ATP is abundant in a muscle cell, it means you're not using it very much. I don't need to degrade my glycogen stores right now. In that case, my phosphorylase in the muscle will be less active. If ATP is diminished in a muscle cell because you're running or exercising or using it, then a lack of ATP is indicated by a higher concentration of AMP, which means you've lost some phosphates, you've used them. So in that case, AMP activates the phosphorylase in the muscle cell. In the liver, it's quite different. It really doesn't detect the amount of ATP as much, and I'm more concerned with the concentration of glucose from the blood. So if the blood glucose drops, I want to degrade glycogen, so it's being more active. If the blood glucose is high and glucose is available and it binds to this thing, then maybe we don't degrade it right now because the blood's happy with the amount of glucose it has. So I want you to know the difference in the two forms, not necessarily all the other A's and B's and tense and relaxed states. I don't care if you know those. I want you to know there are two forms of phosphorylase, one that mainly exists in muscle and one that mainly exists in liver. And the one in the liver is sensitive to the amount of glucose in the blood, and the one that's in muscle is sensitive to that muscle cell's ATP concentration. Okay. So that's what this slide is describing. And I just want you to look at the picture on the right. At the top, in the muscle, you notice the binding of glucose is what tells this thing to become active or less active. And ATP, at the bottom of the slide, phosphorylase A in liver, the binding of glucose decides if this thing is going to be active or inactive. So if glucose were to bind, then it's abundant and I don't need to be as active right now. But if glucose is absent, It won't be bound in those sites, and the enzyme becomes more active in that R state. In the top picture, in muscles, it's not so concerned about the amount of glucose, but it's concerned about its own well-being and the amount of ATP it has. So it'll either have ATP bound or a lack of ATP bound, which means AMP will bind. It's the same thing I just said on the previous slide, just shown in pictures. The phosphorylase in both cases, in liver and muscle, has sites to bind to ATP or AMP and glucose, but one is preferred to be the trigger in each of the two enzyme types. Okay. So how does this thing get activated? Right, so this is the one in the muscle where we're going to have ATP binding and it becomes active, right, or inactive. So if this thing is to work, we need to put a phosphate on it, right? So you notice the little P in the picture on the right. It goes from phosphorylase B to phosphorylase A. It needs to get a phosphate put on it. What would put a phosphate on a protein? Well, there's an enzyme class that does this. They're in the class of transferases. And when you transfer a phosphate, this is a subcategory called kinases. We talked about these before. So kinases are just a specific form of transferases where they transfer phosphates. So if you transfer a phosphate, you're a kinase. And from where do you get your phosphate? Usually an ATP or a GTP or some other source of phosphate like that. And the enzyme that do it, in this case, is called protein kinase A. A very simple name. It reminds you that it's a kinase. And protein kinase A phosphorylates numerous things in the cell. And it depends on the cell type and what's available. It's kind of like Oprah Winfrey, right? So a kinase will give away phosphates to everybody, just like she gives away cars to everybody. So it's everybody gets the phosphate if you're a kinase. If you have a site that is accepting of a phosphate, a kinase will put it on. Right? But that kinase has to be activated itself. So in this case, we have a fully active kinase shown on the right with the phosphates attached and the calcium attached and everything else, the little green and white shapes. But for it to be active, it had to be phosphorylated itself. Okay, so for this enzyme to work, protein kinase A had to put those phosphates on glycogen phosphorylase. Now I know it sounds like I just said the word phosphate or phosphorylase 7 times in a row. So let's clear up what these words mean. On the far right, we have an enzyme. It's shown by the gray and yellow blobs. That enzyme is glycogen phosphorylase. It was step 1 of degrading glycogen. That's its job. But for it to work, it needs to be phosphorylated. So an enzyme that phosphorylates something is called a kinase. Okay? A kinase puts a phosphate on something. So the little green and white picture here, right in the center of the picture, is an enzyme that puts phosphates on this protein on the bottom. Okay, that protein's name, again, was glycogen phosphorylase. So if you were to name an enzyme, let's step away for a second and just name an enzyme that puts eyelashes on people. Or maybe there's an enzyme that you could imagine doing that. You walk into a place, it puts an eyelash on you. Right? You would name that enzyme by its substrate name and then its function. Right? So it'd be the, we're going to make up a name for something that puts eyelashes on people. We'll call it a lashase. It's a silly name, but you'll remember it. And what does it do? It puts eyelashes on things. What people does it put it on? It only puts it on people who are over 6 feet tall. I'm just making this up. Right? So we need a name that describes its substrate. So it'd be the 6-foot lashase. So it describes only those substrates with which it will interact. So in our case here, we have protein kinase A that puts phosphates on many things. That's why it doesn't really have a first name. But the one that's now active here, the green and white thing with the 4 phosphates on it and the calciums, is a very specific kinase. It puts phosphates only on this protein on the right. So its name would be the phosphorylase, that's its target, kinase. That's what it does. So the one on the right is called phosphorylase kinase, and it phosphorylates phosphorylase. I know that sounds confusing. It sounds like the same word over and over. All right, so make sure you get it straight. Phosphorylase kinase, which is this thing here, right, is activated by 2 things. It needs calcium bound to it, and it needs to be phosphorylated itself. And that's done by protein kinase A. Okay? Protein kinase A is activated by a cascade, which we won't go through all the cascades and transmembrane receptors and all that, but know that the picture at the bottom is what I want you to get out of this. Okay, you can ignore the rest of the slide if you want. We'll look at the bottom. We have to tell this cell to do this. So let's say this is a muscle cell. And your muscle cell is full of glycogen, resting and not doing anything. And suddenly you're chased by a bear. Right? You need to get away. Your muscles need to work. You need to mobilize glucose so you can keep running. Right? That extra few seconds might save your life. So your body needs to tell that muscle cell, in anticipation of this, within seconds, release your glycogen, right? Let it go. Let the glucose come out of it. Break it down so you can make more ATP rapidly. So how does your body tell your muscles to do that? You release a hormone, right? And in this case, the hormone will most likely be adrenaline or epinephrine, right? You can make it a little more tame example or equally dangerous example. You're driving in Atlanta on 285 and you almost get run over by a truck, right? You're going to feel that excitement in your muscles, right? They're primed to contract, right? They're ready to go. You have to pull over and listen to your heartbeat for 5 minutes. So that epinephrine is released. It binds to a receptor on your cell membrane on the muscle, and you see this large cascade of events going on. But what I want you to notice is at the very bottom is we activate protein kinase A. Protein kinase A does what? It does what kinases do. It puts phosphates on things. So it phosphorylates the protein called phosphorylase kinase. Well, that phosphorylase kinase, as you see in the picture, goes from red to blue. Blue meaning it's active in this case. It's now an active kinase. Well, phosphorylase kinase is a kinase. It does kinase things. It does what kinases do. It puts phosphates on things. And this one in particular puts a phosphate on glycogen phosphorylase, making it active from the B to the A form down there. And when it's active, it breaks down glycogen, which was our goal. So why the long cascade? Why all these steps? Why not just have epinephrine bind and that activate glycogen degradation? The beauty in having all these steps is you get an amplification at every step. Each of these enzymes turns on thousands of other enzymes. And each of those activates thousands of others. It can do it over and over and over. So because of this cascade, we get a huge amplification of the signal. Okay, so one epinephrine molecule binding can turn on millions of enzymes. And that's why it's so rapid. You can get one binding and then instantly degrade most of the glycogen in the cell, right? Or a large proportion of the glycogen in the cell. So that muscle's ready to go. Okay, so we don't want this to go on forever. If we degrade all our glycogen and keep this process on all the time, how do we ever stop and build more glycogen? Well, that phosphate I put on with that glycogen phosphorylase, let me go back to that one here. The glycogen phosphorylase here gets phosphorylated, right, to make it active. Same thing shown here, from inactive to active, right, in our muscle cells. I don't want that to stay on forever. To turn it off, I need to remove this phosphate. I need to cut it back off. Same thing here. I go from phosphorylase A in blue back to the red in B here. So that needs to be turned off. How do we do that? Well, I just simply cut the phosphate off. So I cut it off by using this enzyme called protein phosphatase 1, right? Which is a hydrolase. And just like the enzyme that exported the glucose for me, or made it possible to export it in liver cells, here we're just going to cut off the phosphate. We're going to add a water molecule and cut it off. And this thing is always active in the cell, constantly. It always cuts off phosphates. If I really want to turn glycogen phosphorylase on, I'm going to have to compete with it and put phosphates back on all the time. Well, I'll keep doing that as long as I need. And then when I stop needing to put phosphates on, the phosphatase will eventually win. Yes, it's a little wasteful to keep cutting it off when I want them on, but it gives me a way of having an automatic off switch. It turns it off when I'm not using it. It's a default case to turn off. So if you wait a while, or you're climbed a tree to get away from the bear, or you're pulled off onto our exit ramps to avoid getting hit on the highway, you have to sit and let yourself calm down for a while, because you got to take time to turn this process off. It's not immediate. If you've ever been scared, or suddenly someone frightens you, and it takes a few minutes to calm down because you've activated the system and it's not an immediate off. Okay. In the other pathway, let's say we're not exercising or running from an animal that's trying to kill you or eat you. Let's say you just ate a meal, you're relaxing, you'd like to take all this excess glucose you have in your body right now, in your diet, and convert it to stored glucose in the form of glycogen. So this is a after a meal type thing where the glucose has entered the cells and we want to store it as glycogen for later. So how do we do that? Well, it's almost a reverse of breaking it down. So we're going to start with glucose that you just ate. Glucose goes through the first step of glycolysis. This is not shown on the slide here. We already covered this. Glucose gets phosphorylated by hexokinase to glucose 6-phosphate. And then we convert glucose 6-phosphate to glucose 1-phosphate using the very same enzyme I talked about earlier. And why will it go in that direction? Because there's lots of glucose 1-phosphate, lots of glucose 6-phosphate around and very little glucose 1-phosphate. You just had a whole meal. And you're well fed, there's lots of ATP, there's no need to degrade it right now. So the idea here is we force it to go the other direction and turn the glucose 6-phosphates into lots of glucose 1-phosphates. At that point, we haven't used any new enzymes that you haven't heard of. But now we're going to use a couple enzymes to make glycogen. So we're going to take glucose 1-phosphate, like we just talked about how it's made, and we're going to add a molecule of UTP. This is a cousin of ATP. Right? There's UTPs, there's GTPs, there's CTPs, and ATPs, right? So here's a UTP, it's a uridine instead of an adenine, that's the only difference. And it still has its 3 phosphates. You see the first one in blue, the alpha one, and then the beta and gamma in black. Okay? So if you add up the phosphates in this picture, I have a glucose 1-phosphate, which contains 1 phosphate, and I have a UTP, which contains 3 phosphates. There's a total of 4 phosphates in this reaction. There better be 4 at the end of the reaction. We fast forward through the enzyme and we get a combination. You notice the 2 black phosphates leave, the beta and gamma phosphates of UTP, leave as pyrophosphate, that's the PPI written on the right. And then we're left with the connection of the other 2. The pink one or red one is connected to the blue one. So we have a uridine still there with 1 phosphate left, and we have our glucose with 1 phosphate still on it. But the way we name it is we name it as a glucose attached to a UDP, even though that phosphate didn't come with the uridine. We just name it that way. But you're right in that there are 4 phosphates still there. You see the 2 in the UDP-glucose and the 2 that left as pyrophosphate. So don't think we gained a phosphate or we lost just 1. We actually lost 2 off of the uridine, and there's still 2 remaining where the 2 were synthesized together, put together, fused. Okay, so we start with a glucose 1-phosphate and a UTP, 3 phosphates there, and we make a UDP where 2 of the phosphates were bolded together, and then we have a pyrophosphate that left, both of which came from the latter part of UTP. Right, why do this? Is because UDP makes an excellent leaving group in a second. So we're going to use that leaving group as a means to connect this glucose to the next one. A phosphate alone was not a good enough leaving group. We had to make it a better leaving group. So at the expense of a UTP, we put this on here so that we can attach this glucose to the growing chain. Okay, so this was a simple UTP plus glucose 1-phosphate to make UDP-glucose. We use it in the next step. We take our UDP-glucose and our glycogen chain that's just sitting there. Here's that other figure I was talking about with the square corners underneath. I didn't fix these like I did the others, but these should also just be straight lines. Don't think of it as an extra carbon. It's not. They're just indicating that these were down. Right? And we're going to connect this last sugar in this long chain to the new sugar, the red one that's entering. So the red one gets connected to the growing chain, and you notice that the UDP is a leaving group. It's an excellent leaving group. Okay, so now I have a glucose chain, a glycogen chain, that is 1 glucose longer. So it's just the opposite of the other process. You notice the glucose comes in with a phosphate, we put it on UDP, and then UDP leaves with all the phosphates. So our glycogen in the end has no phosphates attached, just like how we started. Okay, and we can keep doing this over and over and over, add as many phosphates or as many glucoses to the chain as I'd like. But if I only do that, I'm only going to get a really long chain. It's going to make a really long set of sticks. They're not going to be connected to each other. So the only other thing I need to do to get back to the original ball of glycogen is to branch this thing. Okay? Well, when we debranched it, we had an enzyme called the debranching enzyme. So very conveniently, we have an enzyme here called the branching enzyme. Okay, so we're going to take the, oops, I skipped it. We'll come back to that slide in a second. I want to show you the branching enzyme. So if you look at the bottom picture, we have our core. We have several sugars in yellow here attached. We can keep adding more and more and more sugars, so that's what we've done below. I've added quite a few of them, some in yellow, some in blue. No difference, it's just that's how many I've added. So I've got a really long chain of them now. And I can make this as long as I want, but at some point I need to branch this thing. So this branching enzyme typically comes, and we're going to say it always does to make the math easy for you. It's going to grab the last 7, so they're highlighted in blue. It's going to pick up the last 7, pluck them off of the end, and reattach them somewhere else on the number 6 carbon. So it breaks a 1,4 bond and makes a 1,6 bond. Right? What kind of enzyme would the branching enzyme be? What class? So I took it off and then I reattached it.
Isomerase.
Exactly. So it's just like the debranching one where we moved it around, right? But that one also had a piece where it cut it off. So it wasn't technically just an isomerase anymore. This one, that's all it does. It doesn't cut anything off. It just moves that piece. So be careful when you hear questions about this. And the verb is very important. If they say what removes, what moves, and what adds. Those are all different things. The glycogen synthase does a lot of adding of sugars, right? It puts them together. The rearrangement here, the branching enzyme, just moves things. It doesn't remove anything. Whereas the debranching enzyme and the glycogen phosphorylase that tears it apart actually remove things. They permanently break bonds and don't reattach. So be careful when you read the questions on what verb it says. Move, add, or remove. Those are different things. And so I can keep doing this. Now that I have it branched, I can add on to the blue and yellow ends again. And then when I get enough where I can grab 7 more, I can branch one more time. And this can get quite complex and I get that big ball I started with. Okay. But what if I want to start a brand new glycogen molecule, right? Not one that already has this core and a few pieces on it where I could add to. I want to start one from scratch. Okay, so what if I want to start a brand new one here? So I start with that little protein I talked about, that glycogen, right? Represented by the little yellow G here in the center. Okay, so it's a little protein, and it also acts like glycogen synthase does. It has the ability to add sugars to itself. So if you can imagine you being the glycogen, and you pick up a glucose molecule, right, one of these little hexagons, and you stick it on your shoulder, right? You pick up another one, you add it onto the end of that glucose. You pick up a third one, you add it onto the end of that one. You could actually add a whole bunch, but at some point you won't be able to reach the end of the chain anymore. It'll get so long that your arm won't reach the end of the chain, right? So this thing can't put them on indefinitely. It can put on about 4 to 8 sugars, right? 4 to 8 glucose molecules, and that's as far as it can go. We call it auto-catalyzing the attachment of glucoses to itself. It most definitely puts on the first 4 because our glycogen can't get that close. Our glycogen synthase can't get that close. But it can also put on numbers 5, 6, 7, 8, and it can't quite reach far enough to put on the 9th one. Okay? The glycogen synthase, as you see on this slide, can put on, if there's already at least 4 there, it can add more. It can't get any closer than that though, right? So it can't put on number 1 or number 2 or number 3. So there's an overlap here. The glycogen, the original molecule, can put on the first 4 to 8 sugars, so numbers 1, 2, 3, 4, 5, 6, 7, and 8, and then the glycogen synthase could take over anytime after the 4th one's there. So it could put on number 5, it could put on number 6, it could put on 7, 8, but we know for sure it put on number 9 onward, if that number scheme makes sense. So there's a little overlap of when these are built from scratch. The glycogen definitely puts on the first 4, there's no doubt. Who put on numbers 5 through 8 could be either 1, the original core, or the glycogen synthase, but number 9 onward was most definitely put on by the synthase, not the core. So there's a little overlap there. Right. So how do we control this one as well? We don't want to build glycogen inappropriately. If you're in the middle of running from the bear, you don't want to be building glycogen, you want to degrade it. So we need some kind of control over these things. If I'm building glycogen, I don't want the degradation process going on. And if I'm degrading it, I don't want the building process going on. I need some way to control both of those. And ideally, I'd like to have it in one signal, right? So one single signal tells me to do one or the other. And that's what I'm going to show you in the next slide. You don't have to worry about the numbers here so much, how efficient this thing is. The only comment I'll make about that is the more you branch it, the less efficient storage it is. Because remember, every time you branch it, and when I degrade it later, I don't get back the glucose with the phosphate. I get a plain old glucose from that branch point. So I'm down one phosphate every time I branch. But the more I branch, the more compact it is, so the better storage. So there's a trade-off. Right? So I want you to remember from that. Okay, so here's my cascade one more time. Right? And again, let's look at it if it's a muscle cell. Epinephrine's released. We have this large cascade of events. You can fast forward all the way down to the blue protein kinase A. And you've seen the left side of the figure already. Protein kinase A does what kinases do. It phosphorylates, and I said it phosphorylates a number of things. It's not so specific. And one of the things that phosphorylates in a muscle cell is phosphorylase kinase. Phosphorylating phosphorylase kinase, making it from the red to the blue here, are active, turns it on. And of course, it is a kinase, so it does kinase things. It phosphorylates something, and it phosphorylates glycogen phosphorylase, as it says in its name. Phosphorylating glycogen phosphorylase, making it from the red to blue here, turns it on. And we already talked about what glycogen phosphorylase does. It's that first step in degrading glycogen. It cuts off that last glucose. It uses phosphate as a salt. That's why it's a phosphorylase and not just a generic hydrolase. And we get a liberation of glucose 1-phosphate I can use in glycolysis. So this is clearly degrading or mobilizing our glycogen for use, for fuel. At the same time, I want to make sure I'm not trying to build a glycogen molecule, because that would be counterproductive. So back to protein kinase A, I wanted the same signal to tell me both things. So this is necessarily how it has to work. A protein kinase A, again, is a kinase. It phosphorylates a number of things. One of the other things it phosphorylates is glycogen synthase. Well, glycogen synthase, normally not phosphorylated, is active. But when you phosphorylate it, it becomes inactive. So it's a reverse as the other one. Here, our phosphorylase kinase and our phosphorylase both become active upon phosphorylation. They get a phosphate stuck to them, they become active. Here, if you put a phosphate on it, it becomes inactive. And you probably can see logically why it had to be the opposite scenario, because I wanted one signal to turn on the degradation and off the synthesis. If they both were turned on by the same signal, they would be competing in reverse reactions. So I don't want that to happen. So phosphorylating this side degrades glycogen, and phosphorylating the synthase stops the synthesis of glycogen. Now let's say the danger is past, the epinephrine or the adrenaline is gone, we shut down this whole system, our protein phosphatase 1 comes along and cleaves all these phosphates off, returning everything to the left side. And then our phosphorylase would be turned off, our phosphorylase kinase would be turned off, our protein kinase A would be turned off, and so we stop degrading glycogen. However, our glycogen synthase gets turned on when that phosphate is removed, and so we go back into the state of let's store glucose as glycogen for later if it's available. So this is after you finish getting chased by the bear, or you almost got run over on the highway. You stop at a restaurant to eat, or stop at wherever you're in the woods to eat. And then you get a lot of food and you want to rest because you had a long day getting chased by a bear. So now you want to start storing all this excess glucose as glycogen, not degrading it. So all this stuff gets dephosphorylated. The glycogen synthase goes back to the active form, and your liver and muscle cells start replenishing their supply of glycogen stores. In the case of the athlete that was running that long marathon, right? After the marathon's over, they're going to be quite hungry. They're going to need some glucose after this, right? So they usually have a meal of carbohydrates like pasta or something. You also did that prior to the race as a glycogen storage measure. Most football teams do this. The night before a large football game or a very strenuous activity, they usually carb load, where they eat lots of pasta and other carbs and breads and things like that, so that they have a large glycogen store for the next day.